Hello and welcome to the Evolution Summer Review for IB Biology. We're going to be going through about a quarter's worth of material here. Alright. First question is, what is this bird? Something we should be able to answer. If you said Darwin's medium ground finch, you'd be correct. It's from the Galapagos Islands. So natural selection. Natural selection is the main mechanism of evolution, and it was first explained by Darwin. That's correct. So it's a paradigm shift that replaced Lamarckism. And so using his data from his travels and from studying pigeons and dogs when he got back home, Darwin created the model of natural selection, so the explanation for natural selection. And what he said is, for natural selection to work, you must have variation. What are our sources of variation? Well, the main source of variation is going to be mutation. So mutation is going to generate new alleles, and then those alleles will be sorted by sexual reproduction, creating new combinations of alleles and increasing variation in a population. There are two main factors that cause natural selection, and that all has to do with carrying capacity, the maximum amount an environment can support of a population, and those are overproduction of offspring and competition for resources, such as food, shelter. Now, in addition to biotic or density-dependent factors, there are some abiotic factors that can affect survival as well, such as temperature, salinity tolerance, pH tolerance, and these can lead to natural selection too. These are typically density-independent factors. Alright, so you have to have variation, and then if there's overproduction of offspring or competition for resources, natural selection will happen, which leads us to survival of the fittest. So this says those with the most advantageous traits are more likely to survive and reproduce. They pass on their heritable traits to their offspring, and over time this causes change in a population. And so evolution is the change in heritable characteristics over time, and the main mechanism is natural selection. Now, it's not the only mechanism. There's another way, besides just random changes, that can happen, indicated by this picture here. You should be able to identify this fish. This is Endler's guppy. So if you said Endler's guppy, you're correct. And it, like the bird of paradise, are an example of sexual selection. Sexual selection is when certain characteristics are signs of overall fitness which affect success in attracting a mate. And so you can see the bird dancing down below. They had the bright feathers and they had their special dances. And so these are signs of fitness, so they're not necessarily more fit, but they are an indicator of that fitness level, such as the baboons, the geladas, and of course Endler's guppies with their spots. Now, is sexual selection always the most important? And the answer is no. So you have natural selection versus sexual selection. And so predation by strong predators will lower sexual traits. This was proven by Endler with his guppy research, where he found that the number of spots diminished over time when there was stronger predators. All right, that's section 1. Section 2, A41. Start with this picture here. Should be able to identify this. It's correct, this is a chimpanzee, not to be confused with a bonobo. Now, evolution. Evolution is the change in heritable characteristics of a population over time, and it has many forms of evidence. first one is DNA. The strongest piece of evidence is DNA, RNA, and amino acid sequence, and you compare for similarities. The more similarities, the more closely related they must be. And since these are passed down from ancestors, you can see that they must be related. Homologous structures are our second main piece of evidence, and these are things that have a similar structure but different function, and they show that divergent or true evolution from a common ancestor. There are two examples you need to know. The first one is the pentadactyl limb, as seen to the right, and the other one that we sometimes forget about is embryos. So embryos are another type of homologous structure. They can also show structures that are only found in development, so they can be useful for tracing evolutionary history that way. They're similar to vestigial structures, which are a type of homologous structure that no longer has a purpose. So homologous structures are a similar structure with different function, and our vestigial structures are from an ancestor but no longer have a function, like a whale's hind limb, your appendix, or your tailbone. Fossils show transitional forms. In the fossil record, we can see how the ancestors of dinosaurs split to become birds, and how we got land animals by finding fossils that show the transition from being a fish to a land animal, or from a land dinosaur to a bird. Our next piece of evidence is selective breeding or artificial selection. This is selection done by humans on a species, and this is when we artificially choose the traits. We can see the changes happening very rapidly over time. All right, in this picture we have a shark and a dolphin. This is showing a different topic, which you should be able to identify. That's correct. It's analogous structures or convergent evolution. So they evolve separately through convergent evolution. So they're converging to look the same, not because of a common ancestor, but because they live in similar environments. So even though the shark and the dolphin kind of look alike, they are not related. Okay, speciation. Speciation is the splitting of pre-existing species. All species must come from a pre-existing species, and it requires two things. It requires reproductive isolation and differential selection. So they have to have different environmental pressures that are going to cause them to shift. Reproductive isolation can come in many different ways, and if you're an HL, we'll cover it on the next slide. If you're an SL, you need to know, and an HL, you need to know the example of the bonobos and the chimpanzees that were split by the Congo River and experienced very different selective pressures depending on which side of the Congo River they were on. HL, so we have sympatric versus allopatric speciation. And so allopatric speciation is when they speciate separately and then they are brought back together and cannot interbreed. Sympatric is when they speciate in the same area and are speciating because of some sort of other reproductive barrier. So geographic isolation is the most common. Behavioral isolation is another one where they have slightly different behaviors, so they don't recognize each other and they stop mating, and over time they accumulate enough differences to be a separate species. Temporal isolation is when the timing for reproduction has been shifted or active time has shifted so that they no longer interact and cannot interbreed. And so those are the three main ways of causing reproductive isolation. All right, next up we have our next concepts. Actually, two concepts. So we have one on the left and one on the right here. On the left, we have adaptive radiation. So adaptive radiation, like the Hawaiian honey creepers on the previous slide, or the picture of the silver swords of Hawaii to the right here, are when a species radiates or splits into many different species due to vacant niches in the ecosystem. And they can coexist due to a lack of competition. So because of these empty or vacant niches, it allows them to speciate rapidly, and they can coexist because they're not competing for resources. Another type of rapid speciation is polyploidy. So that was the picture on the right on the previous slide, was knotweed, which is an example of a polyploid. And so when plants form hybrids with other similar species, they can create a different number of copies of chromosomes. And so almost all animal species have 2 copies of each chromosome, so we're considered diploid. In plants, they can be tetraploid, diploid, triploid, hexaploid, octoploid, decaploid. So they can have 4 copies of each chromosome. They can have 6 copies, 8, 3, and those typically are those are all polyploids, and so they rapidly speciate, and this happens partially because plants can asexually reproduce and then form these hybrids. So most organisms are diploid, but polyploids have more copies. All right, so how do organisms prevent hybridization? You have your prezygotic barriers to reproduction. So you got temporal, behavioral, geographic, mechanical, kinetic, and incompatibility. So temporal is when they mate at different times. Behavioral is when they don't recognize each other as mates. So these are very similar to the reproductive isolations. Reproductive isolation happens prior to becoming separate species, and then these can turn into barriers that prevent hybridization as well. Mechanical, the pieces don't fit together, and then the last one is the gametic incompatibility, so the sperm and the egg do not fuse correctly. Then you have your post-zygotic ones, which there are 2 main ones we care about. Those are hybrid inviability, where the hybrid does not survive or does not make it to reproductive age, and then hybrid sterility, where the hybrid might be viable, but it cannot reproduce. All right, A31. Species. We have our giraffes here. Why do we have our giraffes? Well, what species of giraffe is this? It's a good question. So we have different ways of defining a species. There are 3 main ones we need to know. The first one is morphological species, where we group them by shared traits. So if they share traits, they must be a species. This is the oldest one by Linnaeus and is the least accurate. Then we have the biological species concept, where the ability to interbreed and produce fertile offspring is how we define a species. Emphasis on the fertile part of the offspring. The phylogenetic species concept is our newest one, and it's using evolutionary relationships and DNA evidence to sort them into separate species. So DNA is more accurate, but it can sometimes be difficult to determine where to do the cutoff. So we name species using binomial nomenclature. Remember, it's genus species, capital for the genus, lowercase for the species, in italics, or underlined if you're handwriting. Now the giraffes are on here, remember, because it can be difficult to distinguish between populations and species. So populations are groups of organisms in the same area at the same time, but how do we know if 2 populations are the same species or separate if they never interact? That's where it can be really challenging. The morphological species says, oh, if they look close enough, they must be. But the biological one is hard to apply to populations that are in different areas because they are not producing fertile offspring because they don't interact. But what if they brought them together? Then would they? Well, then, even if they do produce fertile offspring, if you bring them back, if they never would, are they still separate species? And then with the DNA, it's okay, how many differences are enough to constitute a new species? Or is that difference just because there's variation between individuals? So defining species can be really challenging. This is an image of something. You should be able to identify this and figure out what's going on here. This is a karyogram, so it's a set of chromosomes. The first thing you want to do if you see one of these is count up how many pairs there are. So there are 23 pairs or 46 total chromosomes, indicating that this is a human. And then you want to look at the last pair and see those are the sex chromosomes, and you use that to decide, okay, is one bigger than the other? And if the answer is yes, then it's a male, and if they're the same size, it must be a female. Now, there's also karyotypes shown in the bottom right. So a karyogram is the image, whereas the karyotype is the image with the banding pattern and the specific locations of the genes, so that it's a more detailed investigation. Remember, we should be able to decide where chromosome 2 came from in humans. It's a fusion event of 12 and 13 in the primate lineage. So numbers 12 and 13 fuse together to form chromosome 2. That's why they have 48 pairs and we have 46. Genomes. Genomes are all of the genetic information of an organism. It's all the genes, and the sizes vary widely. There is some relationship between size and complexity of an organism. On average, it goes off, but it is not a like one-to-one or a perfect relationship. There are some complex organisms that have relatively small genome sizes, and also some really complex organisms with not that large of a genome. But for the most part, there is some increase in size and complexity. Eukaryotes are a giant domain that has a lot of variation in genome size. There's more difference between species than within a species, though, in terms of genome size and DNA sequence. And so what can we do with this information? We can sequence genomes, and we can use this for better research and for personalized medicine. HL, this is bacteria or prokaryotes. And prokaryotes are even harder to distinguish as species because it is difficult to tell if they can reproduce with each other because they reproduce asexually, and they can transfer genes between each other through horizontal gene transfer. Now you also need to know that chromosome number is a shared trait in a species. So if you're part of the same species, you have the same number of chromosomes. Now, dichotomous keys are used to identify species. So you'd look at this one here and you'd be like, has black feathers. Oh, it must be some sort of like penguin. Oh, but then you'd be like, has an orange beak. Yes, it's a puffin. Or no, it's a penguin. Right? So you do a series of yes or no questions to identify it. So these are puffins. The other way you can do it is DNA barcoding to identify species rapidly. So you take environmental DNA, such as water samples, soil samples, or poop samples, and you can use it to identify the species and what the species has been eating. A32 for HL only. We have here our cladogram. So cladograms, remember, are used for sorting species. What would the closest living relative of a wolf be? Trick question, kind of. It's a leopard and a house cat because they both share a node. And so oops. Oh, come on. Oh, it doesn't want to go over. Anyway, so that right there is the node. All right, classification. So in classification and cladistics, the old method, we had kingdom, phylum, class, order, family, genus, species. In our new method, we use our unranked clades. And so these are our groupings that follow naturally. And follows our evolutionary relationships. So the sizes can vary depending on the group itself. And so clades share a common ancestor and they have shared characteristics. Even though the characteristics aren't necessarily how they sort it, they use the DNA sequence, but because they're ancestry related, they have those shared characteristics. So like all mammals have like the pentadactyl limb, so even though it's been modified, they still have those characteristics. Remember, you need to know your parts of a clade, so you can see here. The root is the common ancestor. A node is where a speciation event happened, and then the terminal branch, highlighted in green and in blue on this diagram, show evolution of a single species. So these are the unique histories of those species. Our molecular clock. The molecular clock is how we identify the time of divergence for species. And so we use the mutation rate and the number of differences between species to determine how long they've been separated. Now, mutation rate is affected by generation time. So the shorter the generation time, the higher the mutation rate. The larger the population size, the larger the mutation rate. And then the selective pressure, negative mutations will be removed, and so we won't see those as part of the mutation rate, and positive ones will be increased by selective pressure. But on average, selective pressure will lower the mutation rate because it's going to make it seem like there are less mutations than there really were, because most mutations are harmful, so they will be removed. All right, what do we use all this information for? We use it for our RNA in the domains. And so it was done by Woese, and so he regrouped and split the bacteria, archaea, and eukaryotes into the 3 domains. So this is above kingdoms. There used to be 6 kingdoms, and now it's been split into 3 domains, with eukaryotes having 5 kingdoms. Okay, C41. What is this SAT? So we must be talking about a population, same species, same area, same time. And so populations, we can count populations. There are 2 methods for counting populations. Those are for sessile or non-mobile species, we use random quadrat sampling. And for mobile species, we use capture, mark, release, recapture, just like it sounds, and then the Lincoln index. Remember to wait a little bit of time after you release them before you recapture them, and to make sure that whatever you've done to mark them does not affect their viability, right? And so doesn't affect their ability to survive. Now, we also have our population growth, and so populations will grow until they reach carrying capacity, and so this is the amount that an area can sustain with its resources. And so this is the population size that it stabilizes around, right? It will fluctuate around it, go up and down a little bit, but it'll be around carrying capacity. And so this is caused mainly by negative feedback control. This is where density-dependent factors such as predation, competition, and disease lower the population size and bring it back towards carrying capacity when the population gets too large. Abiotic factors will cause it to fluctuate more dramatically, so like big temperature swings, droughts, etc. Those ones will affect larger numbers, and they won't be as towards carrying capacity as density-dependent factors are. Interactions, there's a bunch of different kinds of interactions we need to know. Competition can be either inter or intra-specific. Interspecific is between 2 different species, intra is between the same species. Cooperation is primarily intraspecific, where members of the same species will work together, but there can be some interspecific cooperation. Predation is when an animal eats another animal. Herbivory is when an animal eats a plant. Mutualism is when 2 different species work together to survive, and so they both benefit each other. Commensalism is not up here. It's not as formal that you need to know this one, but commensalism is when one benefits and the other is neither hurt nor harmed. Parasitism is when one steals nutrients from another, so think like leech or mosquito. Pathogenicity is the spreading of diseases, and so when an animal spreads a disease to a human, we call that a zoonosis. But animals can also spread diseases to other animals. Allelopathy is when plants secrete chemicals either through their leaves or through compounds or through the rain into the soil, where they then will fight other plants. And so our example was the black mustard plant. We'll fight off other plants that way. Then antibiotic secretion is also done by plants to prevent damage from bacteria. So they create antibiotics which kill off bacteria. There are two types of population control. The first one is top-down, where the predator controls the population sizes, and the second one is bottom-up, where the nutrient availability controls population sizes. And so these differ depending on where you are in the world and the ecosystem, but it varies. They're typically, most of them are either one or another and not both. But it's become more and more common recently that top-down is more in common than we used to think. Okay, last section. What is this referring to? That's right, it's referring to habitats. And so we need to know the different habitats and biomes and their impact. So the habitat is the place in which a community, species, population, or organism lives. Species are affected by abiotic factors such as the temperature, light, moisture, pH, or salinity of the region. These are all abiotic and are non-living factors. And it creates a range of tolerance like we can see in the graph in the bottom. Remember, coral is our Goldilocks species. It needs very specific things to be able to grow. It needs really clear water with lots of light, so shallow water. It needs it to be pretty salty. It wants it to be warm, but not too hot, and it wants a relatively high pH. Here are two other examples of organisms with adaptations that we are supposed to know. That's correct. On the left, we have our mangroves with their prop roots and their pneumatophore breathing tubes and their salt extrusion from the leaves. And on the right, we have a beach grass, and our beach grass has its underground rhizome to help put it in place. And it's got the really thin, narrow shoots so that it's not blown away by the wind. The other thing we need to know is Whitaker's climate graph. So Whitaker's climate graph is shown here. Remember, the temperature starts from hot and goes to cold, and then they have the rainfall or precipitation on the y-axis. You need to know these different biomes and roughly where they fall on the climate graph. So make sure you know those. So you've got the desert, the grassland, and the tundra along the bottom. Above that, you have the taiga and then the temperate forest. And then at the very top, you have the tropical rainforest. You do not need to know the distinction between a temperate rainforest and a temperate seasonal forest. We'll just, for the purpose of this class, typically call that a temperate forest. Biomes. Biomes are groups of ecosystems with similar communities due to similar abiotic conditions and convergent evolution. So why do rainforests throughout the world look similar? Well, it's because if you have similar abiotic conditions, those different adaptations will still be very similar. And so speaking of those, if you're talking about a hot desert, adaptations there would be things like our cactus and our camels and our fennec fox. So these all have adaptations that help them survive the hot temperature. A lot of our animals that live in the desert are burrowing animals, or they're very light in color. Whereas in the tropical rainforest, they're typically, the plants are bigger. They've got these leaves that slope downward, like called thrip tips, to get the water off them so they don't drown. And they have to have adaptations for growing really tall to compete for light. And then the animals have all different kinds of adaptations. Ones that live near the bottom are typically darker in color, and they are adapted to a warm, humid climate. And that is the end of our first review.